Hi, Professor Stefero here, and this screencast continues the discussion about how science works. We're going to focus in on testing ideas. Testing ideas or testing hypotheses is the center of how science works. If we dig a little deeper at the details, you can see that forming and testing hypotheses can be thought of in two parts: gathering the data and interpreting the data. Now, data comes from observations collected, and these can be from an experiment or in the field. And whether the experiment is in the lab or in the field, the goal of the test, the experiment, is to provide evidence for or against a hypothesis. The goal of the experiment is to try to control all the variables except for the one under study. So what is a variable? A variable is anything that could be changed that may affect the results or output of an investigation. So let's take a simple example you may have done yourself or done with your kids, and that is germinating seeds. We all know it takes light, soil, water to grow plants. Some seeds need light to germinate, others do not. But what conditions could vary the number of seeds and or the growth of those plants from seeds? And that could be our basis for an experiment. So the first variable that we're going to define by using our bean seed example is the control variable. The control variable are the conditions or events held constant so as not to affect the outcome of the experiment. So let's take a look at this picture. How many things could we assume might be a control variable? Well, the container, they're all the same. The amount of light, they're all by the same window. The amount of soil looks about the same. These are all the things given to each container as a way to control the experiment. So light, temperature, the amount of water, all of these could be typical control variables. The second variable is the experimental or independent variable. These are the events or conditions under study that the experimenter varies. So in the case of this experiment, what's being varied is pH. Thus, the hypothesis might be mung bean seeds numbers and mung bean seed growth do not differ at varying pH. And remember, pH is simply a measure of acid or alkalinity. That leads us to the last variable, the things that we count or measure, and that's the dependent variable. Dependent variables are the conditions or events that may change due to the independent variable. So in our example, the number of seeds germinating at different pH is the dependent variable. Here are some examples of independent variables influencing dependent variables. Here we've picked the type of medium that the seeds are grown in as the independent variable, and thus we could measure the rate, the amount of time it takes to germinate in the varying mediums, or the number of seeds germinated like we did with the pH experiment. The things that we count or measure are always dependent variable, and they will be influenced or changed by the independent variable. Now that we have the variables, let's talk about the basic design of an experiment. Generally, experiments are divided into two groups: the experimental group and the control group. The experimental group is going to be the one where all variables are applied to the group, including the independent variable. The control group receives all the variables except the independent variable. So the purpose here is to ensure that the independent variable is leading to the assertion stated in the hypothesis. So recall our example hypothesis: varying pH will not affect the number of seeds germinated. So let's say the experimenter used water at different pH to create the conditions of varying pH. The control would get regular water without any adjustment to pH, while the other plants would receive the varying pH as the experimental group. Now that we have gathered the data, next is the analysis and interpretation of results. Data becomes evidence for or against a hypothesis only when they have been interpreted in a way that reflects what is being tested. Thus, the interpretation will lead to support or opposition of a hypothesis, or inspiration to new hypotheses, or reforming a hypothesis. So let's take an example here. Let's say we're studying the evolutionary relationships between flies, spiders, and lobsters. So here the DNA is collected from these organisms. The A, the T, the G, the C are the molecules that form sequences in the DNA. Next, through a variety of statistical and visual representations, the DNA sequences are analyzed for common sequences between the fly, spider, and lobster. Then, based on the analysis, the experimenter can develop their interpretation of whether that data supports or opposes the hypothesis, whether there's a relationship between them or not. Ultimately, the interpretation is shared with other scientists so they can repeat the experiment, compare to similar work or ideas, and then come to a consensus as to whether the interpretation remains supported by the evidence itself. And that, in a nutshell, is how science works. Lastly, let's compare hypotheses with theory. Remember, the hypothesis is the proposed or tentative explanation for a specific set of observations, and a experiment or individual experiments will have hypotheses. But a theory is different. It is supported by a large and usually growing body of evidence. So an experiment does not lead to a theory, but it may become a part of the evidence for the theory. And here are some of the important biological theories that we will address in class this term. The cell theory, that all organisms are composed of cells. Homeostasis, that organisms maintain an internal environment. The gene theory, organisms contain coded information that dictates their form, function, and behavior. Ecosystems, populations of organisms interact with each other and their physical environment. And then finally, evolution. All organisms have a common ancestor, but each is adapted to a particular way of life. We actually will touch on all of these theories as we move through the class. So that's it for now. I will see you soon. Bye.